Okay, here are my note cards. What if genetic diseases could be removed from your DNA sequence? At first, this seemed impossible, but soon it has become a reality with CRISPR gene editing. This is used by the protein Cas9. So CRISPR is a revolutionary science tool which allows for the editing of animal DNA sequences along with human DNA sequences. And this is used to gain desirable traits by, as I said, using the Cas9 protein. Okay, so according to Integrated DNA Technologies, April 15, 2024, scientists correct genetic defects and introduce therapeutic genes to treat conditions. As someone who's interested in science and has researched many different sources, for example, like Integrated DNA Technologies, I find myself credible to introducing CRISPR. So today I'll be discussing how CRISPR's primary integration into animal has now been integrated into human genetic DNA sequencing and the negative effects of it. So primarily, as I said before, CRISPR was used in animals, but now has been more commonly implemented into humans. So according to the FDA, December 8th, 2023, use of recombinant DNA constructs have been introducing traits into plants and animals. And as I said before, it's for desirable traits such as faster growth and a greater reproductive success. And with these more effective and beneficial traits, companies using genetically modified produce gain a greater income and a greater sales. For instance, Oregon State, February 12, 2019, announced that genetically modified salmon have been engineered with Chinook salmon genes in order to grow twice as fast as the normal salmon. Also, CRISPR has been known to make animals resistant to infectious diseases. For example, Biovenic 2026 has an example of the PRRS-resistant pig, which has knocked out a gene which viruses tend to infect. Another example from Biovenic are the chickens resistant to avian influenza transmiss. And this has been used by CRISPR technology in order to prevent the possibility of a pandemic. Okay. Like animals, humans also had their DNA moved around by CRISPR. This has also been used to cure diseases in humans. On May 15, 2025, Children's Hospital of Philadelphia said that an infant with CPS1 deficiency has been treated with a custom genetic editing technique, also known as CRISPR. His normal treatment would conclude of a lung transplant, but due to his young age, that was not able to happen. According to the FDA, over 100,000 people suffer from sickle cell disease, and they have had their blood cells modified by CRISPR. Some conflicts of CRISPR, according to Integrated DNA Technology, April 15, 2024, there are many unknown health risks that can occur with editing your DNA sequence that can be passed on for generations. In animals, genetically modified species compete with the non-genetically modified species, and it can cause extinction. CRISPR has had many benefits and disadvantages in both humans and animals, whether that's curing disease or simply increasing the size. Although it has many risks, I believe CRISPR will be integrated more into the future with many new evolutions. So CRISPR's altering humans and future generations one base pair at a time.